I will record all the classes and post them on YouTube, and I'll show you where to find those links. But yeah, so this is the homepage of the Canvas course, very similar format to last semester if you have my class. Right here on the homepage, you've got some links that are helpful, right? To meet your instructor, you can get the course syllabus right here. This week 1 course orientation page, I want you to go to that and just read through it, okay? You can sign up for office hours right here, okay? I have those in my calendar. That's really, you can just hop on and grab a spot if you want to come talk to me about the class, if you have a question for me, if you want to come talk to me about your life, you want to sit down and pray with me, I'm happy to do all of those things with you. And so just sign up. And if none of those actually work, then none of those time slots work, then just talk to me before or after class or email me, and we can figure out another time. Okay, so that's all there. So first spot I want to go is the syllabus. Okay, so here's the syllabus page. This syllabus page doesn't have a whole lot of information. It puts all the assignments there, but really the better spot to go is this link here that says the syllabus. That's the full syllabus, okay? So I'm gonna click there, okay? All right, so just a couple of things. You guys know from last semester, there's the lecture that contains 3 lab sections, right? So you're all in lecture section 3, but then you're also in lab 3A, 3B, or 3C, right? All of those are taught by Professor Mahoney. And they're the different times. They're 8 a.m., 1:30, or 5:30. Okay, so those will start next week, not this week, right? So that's how it's going to be set up, anyways. Not just because we're virtual, but we always don't start lab till the 2nd week, so we're not behind there or anything. Again, office hours here, another spot to sign up for those. Ways to contact me. Final exam is on Wednesday. Okay, a couple of things here. Yeah, this is just something, kind of my commitment to you that I like to read. And really, I'm here to help you in whatever way you need, okay? So that my door's open to you for you to come to me with questions about the class, but also about your life, about your future, about your career, whatever it is that's on your heart and you want to talk about. I'm here for you. So take me up on that if that's something that sounds interesting, okay? All right, let's skip through some of this. You definitely need the textbook here, as well as the access to Master AMP. And for my course, you will need the iClicker 2 remote. Okay, if you had me last semester, you should already have it. If not, you'll need to get one of those for when we start back face-to-face next week. Okay, you'll need the dissecting kit. And yeah, okay, a couple things I wanted to mention. This one right here is an important one, okay? We recommend about 8 to 12 hours of studying per week just for this course. Okay, I know that's a lot. That's a lot of time. But I talk to the students. I talk to all the students, but I talk to the students in particular, the ones that do really well in this class. And I've asked them over the years, like, about how much time do you spend on this class per week? The ones that are getting the high A's, and it's typically right around there. It's typically right around somewhere around 10 to 12 hours of time outside of class studying for this class. Okay. It takes a lot of time. There's so much material in this course. There's so much coming at you so fast that it just takes a lot of time to process and to absorb all the information and to learn it. So that's not like just a gross exaggeration when we have that 8 to 12 hours in there. It is really what I strongly recommend that you spend in this class. Okay, a couple of things about the assignment structure in this course. On most Mondays, you'll have a Master in AMP assignment due. Okay, so it's a 5-point assignment, and it's always going to be based on stuff that we haven't actually talked about yet in class. Okay, the point of those Master in AMP assignments for me is to get you into the book, get you into the material, prepare you for class so you're somewhat ready for the material when we get to class. Okay, if you come into class and you've never heard of the words that I'm using at all, then you're going to be lost. There's no way you're going to be able to understand the concepts that we're going through in class if you're not even familiar with the vocabulary. Right, and so the best thing you can do is to do that Master in AMP, as well as read the pages in the book that I have for you in the syllabus, and I'll show you where those are, before class each day. And the Master in AMP just kind of forces you to get into the book a little bit to do that. Okay, so that's on most Mondays, except the first week, which we have one due tomorrow. Okay, so and they're always due by class time. Okay, so they're always due by 1:30 p.m. Typically on Mondays, but again, this week we have one due tomorrow, Wednesday. Okay, so you can hop on and get to that. I'll show you how to get to that in a second. Okay, and then the only time we don't have Master in AMP on Monday is when we have a TBL day. And we'll talk more about those, but that's a day where you're going to study a lecture ahead of time, come to class, take a quiz by yourself, take it again as a team, and then we'll go through some activities. On those TBL days, we don't have Master and A&P assignments due. So that's the only exception for the Mondays. Okay, so those are typically on Monday as well. And then every Friday is either a lecture quiz that you're going to do on Canvas. Let's do it 11:59 p.m. Friday of that week. And you can always click on the lecture quiz and look at it, and it'll tell you which 2 note packets it's over. Okay, it's always going to be over 2 class periods. And so you can do that for the first one that's due this Friday. Okay, those are timed. So they're open book, open notes, but they're timed. You have 18 minutes to do each one. Typically, if you don't study at all and you just try to go in cold, you're not going to do well on that. You don't have enough time to look everything up. So I highly recommend that you study just like you would for the exam for those 2 sections, and then hop into the quiz and take it, and you're ready to go. Okay, you can't start the quiz and then stop it. The timer will keep going. Okay, so and number 1, always use Chrome as your browser when you're taking a quiz in Canvas, okay, or an exam, because otherwise it'll mess up and you'll actually lose points because the questions won't show up right if you're not in Chrome. Okay, all right. Yeah, what else? Laboratory attendance is mandatory. If you don't show up to lab on a day where we're doing a worksheet, then you get a 0 out of 20 on that worksheet. If you don't show up to lab on a dissection day, then 20 points is deducted from your 100-point practical grade. Okay. Obviously, if emergencies happen, you need to let us know. But if it's just you didn't show up or you forgot, then you're going to have some points deducted for that. Okay, with the lecture quizzes and with the lab quizzes, I dropped the lowest one. Okay, the one exception for that in lab is there's 2 higher point quizzes. There's a 14-point quiz and 16-point quiz. Those ones don't count in the drop-in, but all of the 10-point quizzes do. Same thing with the lecture quizzes. Your lowest 15-point lecture quiz gets automatically dropped. And so that's an opportunity for you. If you missed one or you forgot it or whatever, then that can count as your dropped one. I don't accept late quizzes in either case. Okay, there's 1 practical this semester over the circulatory system instead of 2 like there were last semester. Okay, this here shows you how your score and how your final grade in the class is determined. Okay, what we do for lecture and lab is we combine the points all together, and then your total grade in both lecture and lab is just the percentage of those points that you got. Okay, so you don't get a different grade for lab than you do for lecture. It's going to be the exact same letter grade, but we just assign it to both lecture and lab, and we combine all those points. That the lab typically helps the lecture grade a little bit because it's most time a little bit easier to get a higher grade in lab. Yeah, and that's how I do my grading there. The grading scale, you can see that. Yeah, and so that is pretty much that. Once we get to class face-to-face, I don't allow electronic devices in class. And that's a big reason why when I send out that paper note packet that I want you to work through, or I want you to have with you, you always need to have that note packet with you for each class period. Okay, there's a lot of stuff to fill out, a lot of little activities to do as we're going through the notes, and so you want to have that with you in one way or another. So if you printed it at home, that's fine, but if you ordered it from the print shop, then you can pick it up when you get here. A lot of those are already done. Okay, I'll show you where to pick that up if you, I'll show you where to order one if you haven't yet. And you still have time to order some. Okay, I think that's it for the most part. At the bottom of the syllabus, you'll see the lecture schedule here. Okay, and this tells you the reading that you're supposed to do before each class. Okay, it has it in modules. The textbook's broken up in modules, so that's the way we have it. It has the quizzes, the Master and A&P assignments, has everything that you need to know as far as what's due, when it's due. Okay, after every class, I'll post the video to YouTube and I'll put it in right here. Okay, so you'll be able to access that either right here on this page, or I'll show you another spot where you can access it as well. This is the lab schedule for the semester. Same thing, it shows you the quiz, tells you what the quiz is on, tells you what's going on each week. Okay, all right, let me show you a couple more things. The basics that you want to go to. Oh, again, this course overview page under the first module is a very important one. This has the combined note packet. Okay, so that's the PDF of the note packet. If you want to print that on your own, you can get it there. Okay, again, if you didn't order one from the print shop yet and you want to, you can use this form to do that. Do it today if you want to do it. And yeah, and then this is your team assignments. Okay, so and I'll get to that later, and you'll be sitting with your team each day in class. But we'll get to that next week. Okay, this just kind of shows you the general breakdown of each week in this class. Okay, the thing I wanted to show you again, here's the office hours link. I have the office hours link in a bunch of different places, but you can get it right there too. Sign up for the appointment there. Course schedule, lab schedule. And then I have all the PDFs of all of the lab handouts for the semester right here. And those are in the order that we're going to do the labs. Okay, so if you want to just download all those and print them today, you can, but you will need those printed in lab when you come to lab. Okay, not every one every day, but just the one that we're doing that day. Okay, finally, the last thing I want to talk about is these weekly overview pages. Okay, so for each week, there's a module set up like this. So here's week 1, it'll have everything that's due right here. Okay, and then you can click on the week 1 overview page, and here is just a little chunk of that bigger schedule. Again, you'll find the links to the videos there. Okay, it'll tell you what's going on in lab right here, and then down in this table, it'll tell you everything that is happening or that's due for the week. Okay, you can click on lecture quiz there, and so every week has an overview page like this. I would highly recommend you go to this to see what's going on each week. Okay, last thing I want to show you is how to access MasterNAMP. Okay, you're not going to go straight. You can't just Google MasterNAMP and log in. It doesn't work that way. You have to go through the Canvas course the way this is set up. Okay, so you're going to do that by clicking on this My Lab and Mastering link. Okay, then it's going to take you to it. It's not going to work on my iPad, but it'll look something like this, and that's how you can access the assignments. And again, you can also just go to the assignments link too, and you'll see all these homeworks. You can click on homework 1, and you can click Launch External Tool, and it'll take you to MasterNAMP, and you can complete the assignment there. But that's the one that's due by class time tomorrow. Okay. All right. Any questions about any of that? All right. Okay. So we're going to do a couple of questions here. Normally, oh, Annalise has a question. Go ahead, Annalise. Yeah.
Yeah. Is the mastering AMP due next week on Monday since it's a holiday, or is it still same time?
No, it's another exception. Yeah. That one is due. Yeah. Let's see. Where is that one? We've got it right here. If we go to this one, yeah, week 2. So you can always just see here, week 2. So yeah, that one says Monday. Huh? It's not supposed to be Monday. Yeah, I'll have to fix that because we actually don't have a MasterNAMP 2 next week because we have the TBL day. So there's no MasterNAMP next week. It's the TBL day on Monday. I just forgot to delete this part of in that table there, but yeah. So no MasterNAMP next week because of the TBL day on Wednesday instead. Good question, thank you. All right, other questions? Okay. All right, so we're going to do just a couple of poll questions here on Zoom. And so one part of team-based learning is that when you come to class after you've studied the material on your own on those TBL days, is you're going to take a quiz by yourself, and then you can take that same quiz again as a team. Total between those 2 quizzes is 10 points. And I'm going to give you the option to decide how much of that's going to go to the individual part, which is the IRAT, okay, and the team part, which is the TRAT, okay. So you get the option to vote here. Do you want to put more of that on the individual score or more of that on the team score? Okay. So does everybody understand what they're voting for here? Okay. All right. So let me go ahead and start that poll. And then there's going to be a couple other questions that you can answer as well. You'll see there, and it'll just be on the next slide. More information on that. The first one's what year at Loma are you? And then it goes down from there. Is that working? You guys see in the poll? Yeah, okay. There we go. So yeah, so this question here, yeah, then the other questions are right here. So what year at Point Loma are you? You can see those answers there. What's your major? And then who did you have for Bio 1030 last semester? And you can see those in the note packet too. If you need it, they're not just here on the screen, but they're also in the note packet. And then you can see there's a question about what we just talked about with the syllabus too. That one's also there. So you can take a look at that one. That's in the note packet. If you just flip the page, I think you'll see that as well. Just a little making sure you're paying attention on the syllabus. What's the basic structure of this class as far as when's the Master and AMP due, when are our TBL days, when are our quizzes, when are our exams. Okay. Yeah, go ahead and just go through all 5 questions there. Give you another minute or so to answer those questions. And this is how you're getting your participation points for today, since we don't actually have the clickers yet. We're not in class to use those. There you go, Mallory. All right, we got about 50 of you that have answered out of 69, so we're almost there. Give you about 30 more seconds to complete these questions here. All right, looks like we got just about everybody. Okay, I'm gonna go ahead and end that, and then we can take a look here. So can you guys see those results now? Yeah, they pop up for you? Give me a thumbs up if they popped up. Yeah, good. All right, cool. So you can see that, yeah, for this first one, you chose the thing that every class in the history of me doing this question has ever chosen, which is the most points to the team as possible and the least points to the individual. So that's what we'll do. And then what year at Loma are you? So we've got we've got about 41% of you as freshmen, about 29% of you sophomores, and then we got some juniors and seniors there too. Okay, your major, we got about half and half, not quite half and half, but 40% each in nursing and kinesiology. And then we got some nutrition, we got some biochem, and then we got a few others as well. And then who you had for Bio 1030, so about almost 70% of you had me, and then there's a few there that switched over. Okay, and then lastly, which one here for the syllabus? So 84% of you said C. So Monday is typically our Master and AMP, Friday is the lecture quiz or exam, and then Friday is the TBL day. That's actually not right anymore. We didn't talk too much about that. Pencil's not working. Hold on one sec. Yeah, but so the Fridays are not the TBL days, but that was the one that was closest to being right. I forgot to change that question because last spring the Fridays were TBL days, but the TBL days will be on Mondays, and they'll replace those Master and AMP assignments. Okay, any questions about any of that? Feel free to throw something in the chat or just unmute. Okay. All right, see if my pencil's working now. Good, there we go. Okay, so we're gonna hop right into the material here, if there's no questions on any of that. So yeah, so we left off last semester with the nervous system. Okay, we talked about central nervous system, kind of general basics of the nervous system, the brain, the spinal cord, the cranial nerves. Okay, and just so you know, your first lab quiz next week is over the cranial nerves. Okay, so good to hop back into those the cranial nerve stuff that you learned last semester. And in particular, just the number and name of each cranial nerve, and then what their functions are. Okay, Yeah, and so then this semester we dive into the sensory side of the nervous system. We talk more about the motor side of the nervous system. Last semester, we're going to start out with some general senses today, and then starting on Wednesday, we'll get into the special senses, starting with the eye, then we'll get into hearing and smell and taste. Okay, so this is just kind of a reminder of this hierarchy of the nervous system, where we were last semester. We obviously talked about the central nervous system, talked some about the peripheral nervous system. Again, mostly it was motor that we talked about. We talked about the somatic motor for the most part. So this semester, we're going to talk about the sensory, which is right here, where we're going to start today. But then we're also going to talk about the autonomic nervous system as well. But today we're right here on the sensory or afferent side. Okay, so in senses, there's kind of two broad categories of sensation that we have. Either the general senses, which include touch, temperature, and pain, and these are found all over the body, okay? Or we have special senses, which are driven by these complex sensory organs that are all located in the head, okay? So it's vision, hearing, smell, taste, as well as balance. Okay, so one thing to kind of ponder and think about when we think about sensation is this idea between what's the difference between us sensing something and us perceiving something, okay? So sensation is really an awareness of changes in our body, and those could be internal changes or external things happening that are changing our body. Okay, so that's what sensation is the awareness of that. But then perception is, all right, we've become aware of this, but now how do we interpret that? Do we interpret that as a threat to our body that we need to react against? Do we just kind of ignore it? Do we take some action to, maybe if it's a temperature receptor and it says we're too hot, do we take some action to cool ourselves back down, right? And so there's the perception piece that's very important, and that's going to happen in the brain, in the spinal cord, right? The brain is going to have to decide what to do with that sensory information as far as what kind of motor output to send out to either change that sensory or that information, so the sensory information changes, or just ignore it and not do anything about it, okay? And so these things are so important because feedback is essential for life. Right? Your body can survive in so many different environments externally, and so many slight changes in environments internally. Okay, and so the real bottom line here is that we have to be aware of all of these changes inside of us and outside of us, and then our body has to make changes in order to maintain homeostasis. And so we have to have this feedback, and that's what the sensory nervous system is all about. Okay, a couple of things that are important to understand when it comes to the sensory side of the nervous system is, for one, how does a receptor, first of all, send a signal to the central nervous system? Okay, and I think we should probably know this from last semester, but again, that's going to be driven by an action potential, right? So AP, action potential, okay, and then the way that the brain knows whether that's an intense signal or just a kind of a mellow signal, it's all about the stimulus strength, okay? And that stimulus strength is not about how tall each action potential is, it's about how many action potentials are arriving at any given time, right? And that's the frequency part of that. Okay, so action potentials are frequency modulated. Okay, and we'll talk about that a little more in a second. Okay, so that's kind of how the brain can perceive the difference in the strength of the stimulus is all about frequency, how many action potentials are arriving at any given time. Okay, and then there's sensitivity to a stimulus is going to be dictated by the threshold of the receptor, right? So there's certain insects that when they land on our skin, we don't feel them at all, right? They're just not heavy enough to put enough pressure on those pressure receptors to go above the threshold, and so we don't feel them, okay? But then we put something a little heavy on there and we do feel that, right? So that's that idea of the threshold concept in receptors. It has to be a large enough stimulus to be above the threshold of that receptor. Okay. So this is just kind of a schematic that kind of represents that a little bit. We talked about last semester the resting membrane potential somewhere around -70 millivolts, right, of a neuron. Okay, And then if we can depolarize up to above this threshold of -55, then we get an action potential, right? And so these are representing our action potentials. The red lines are representing the stimulus, right? So if we think of this as a pressure receptor, right? Then this would be a small stimulus. This would be the example of that little insect landing on us that's too light for us to even feel it. Okay, so it changes, it depolarizes the membrane a little bit, but it doesn't get up to the threshold, so we get no action potentials. So we don't feel this one, right? It's a sub-threshold stimulus, okay? But then we get a bigger insect that lands on our skin, and it's big enough now to depolarize the membrane enough to go above the action potential threshold. We can see that here. So we start getting action potentials, right? We get some action potentials. We get an even bigger stimulus, we get more action potentials, right? They're more frequently occurring. We get an even larger stimulus, they're even more frequently occurring, right? And so the brain interprets those different frequencies of action potentials in a different way. So the brain knows the difference between something very light landing on us versus something heavier pushing against us, right? And that's the way that the brain can interpret that, is by receiving a different frequency of action potentials. Okay, questions on that? Okay, all right, so let's talk a little bit about the different types of receptors. So there's two primary categories. They're either called tonic or phasic receptors. Okay, tonic receptors don't adapt. They continue to send action potentials as long as the stimulus is there. Okay, so some examples of that would be nociceptors, which are pain receptors, and proprioceptors, which are kind of our balance receptors. Okay, and this is important because pain's there for a reason most of the time. And so the brain always wants to know about pain, right? We don't, unfortunately for us, the pain stimulus doesn't really, if the pain stimulus is there, then the pain sensation doesn't go away, right? And that's kind of unfortunate for us in some ways because it hurts, but it's also good for us because there's something wrong with our body and we should know about that, right? So those are all tonic receptors. So as long as the stimulus is there, we feel it, okay? But then there's phasic receptors that, we only feel it when there's a change in the stimulus, okay? So some examples of this are our touch receptors, right? When we all got dressed this morning and we first put our clothes on, we kind of felt the sensation of those clothes being on our bodies, but then after a while, we can't feel it anymore, right? And that's good because it would be super annoying if we were constantly aware of every single contact point of our clothing on our skin, right? And so touch receptors adapt, and we press down on them a little bit with whatever's touching it, and we feel that change, but then if the stimulus stays the same, we're not aware of it anymore, and that's called phasic receptors. Okay, we can also adapt at the level of the brain, right? Even if the receptor doesn't adapt, the brain might say, all right, I'm tired of feeling that, I'm just going to ignore it for a little while, right? And the brain can do that as well. A related concept is this idea of what's called a receptive field, which is there's and I'll show it more on this slide, shows it a little bit better. Receptive fields, when we touch something on our skin right here in this blue area, that's the receptive field of one single touch receptor. Okay, so if we touch right here on this touch receptor, okay, the brain doesn't know where within the circle that we were touched. It just knows somewhere within that circle we were touched because that just activates the neuron that's connected to that receptive field. Okay, so then the larger the receptive field, really the less specific the brain knows as to where we're being touched, right? So then in places that have very sensitive sense of touch, like the tips of our fingers, our lips, and things like that, there's a lot of touch receptors, and those touch receptors have much smaller receptive fields, so we get a lot more specific information in those areas. We'll do a nice activity in lab that kind of demonstrates that. Okay, so that's the idea of receptive field. A couple things here as far as receptors go. We can classify all of these different sensory receptors in a couple different ways. Number one, we can classify them based on where those receptors or where the stimulus comes from. Okay, so there's exteroreceptors. Examples of these would be receptors that are receiving information from the outside of our body, right? Externally to our body. Those would be touch receptors or vision receptors, like in our eyes, light receptors. Okay, or they can be interoreceptors, also called visceroreceptors, which is receiving information inside of our body and telling our brain about it, right? So what's the pH of our blood? What's our level of oxygen or CO2 in our blood? What's our blood pressure? Pain that's coming from our deep in our body. Those are all interoreceptors. Okay, we also have proprioreceptors, which are giving us an awareness of where our body is positioned in space. Okay, we have some special proprioreceptors that we'll talk about in our tendons and ligaments, our joint capsules and our muscles. We'll talk about a couple of those at the end of the notes here in this first class. Okay, but then we can also classify them based on the type of stimulus. Okay, so if it's a mechanical stimulus, we call them mechanoreceptors. If it's a temperature stimulus, we call them thermoreceptors. If it's a chemical stimulus that we're sensing, we call them chemoreceptors, right? Light or vision, we call photoreceptors, and then pain receptors, we call nociceptors. Okay, so there's examples of each of those there. Okay, and it pretty much tells you in the name what the receptor is receiving, right? So what is the stimulus? Oops, sorry. What is the stimulus is the key for naming those types of receptors. Okay. What questions do we have so far? Okay. Give you another sec here to finish writing that down. All right. Ready to move on? Your thumbs up, ready? Got it down? Most of you are nodding your heads. Give me a thumbs up. Okay. All right, so we'll talk a little bit about a couple of the specific receptor types and how we sense those. Okay, so temperature receptors, again, dictated by thermoreceptors. Okay, and we have both cold and warm thermoreceptors. Okay, and both of these, so the cold receptors respond to temperature decreasing, whereas the warm receptors respond to temperature increasing. Okay, and we do have kind of these specific ranges. And you don't need to memorize those ranges. It's not that big of a deal there to memorize the temperature, the actual numbers, but just the idea that we're responding to cooling temperatures or warming temperatures. But then the issue with those ranges is if we get outside of that range, we get above it for the warm receptors, then we don't feel it anymore. If we get below it for the cold receptors, we don't feel it anymore. And that's obviously getting into temperature zones that are dangerous for the body to be at anyways. But yeah, so that's thermoreceptors. Pain receptors, or pain is detected by nociceptors or nociceptors. Okay, the different types. The interesting thing about nociceptors is they can be stimulated by many different stimuli. Okay, so we have some that detect chemicals, right? And so those same chemoreceptors that might be telling our brain how much oxygen we have in our blood. That's not a good example, actually. The same chemoreceptors that are detecting our pH and telling our brain what our pH is, if that pH gets too low, that can turn from just a simple stimulus telling our brain what our pH is to pain, right? And so that's what burns when you're exercising intensely as your pH drops a little bit, and that actually causes a burning sensation because those hydrogen ions are stimulating the pain receptors. Okay, so it's the same way with hot, with cold, and with intense pressure or pinching. Those things all lead to actual pain sensation instead of just feeling warm or feeling cool or feeling pressure. They can actually turn into pain when it's a serious or much higher stimulus. Okay, so like we talked about last semester, all of this sensory information is going to make its way up to what's called the somatosensory cortex. Okay, and if you remember where that was in the brain, it was in the parietal lobe of the brain. Right? And we have this little guy, we can put this thing out as a full person called the sensory homunculus, right? I think we looked at that a little bit last semester. And really what this represents is the lateral side of your brain. So down here by your temples, right? Or down by kind of by your ears, whereas this is the middle, this is the, get that thing out of the way. This is the middle part of your brain, kind of right in the center part, right? So all of the touch receptors that go to your toes are found here in that very medial part of your brain, whereas all the ones that are connected to your tongue are found way over there on that lateral part of your brain. okay? And the thing about this is the larger the region in the brain is for each part of your body, all that means is there's more neurons that go to that part of your body. So there's larger area in the brain that makes that up because that's where the cell bodies of those neurons are. And so that's the idea. Again, this information is coming up to the somatosensory cortex and parietal lobe. Okay, so let's we're going to spend the last 10 minutes of class here talking about reflexes. And reflexes connects directly to this idea of sensation because they're always driven by us sensing something. Right? So there's always some kind of stimulus that is being detected, and then that sensation is going to go back to the spinal cord, and then the spinal cord sends out a couple of actions to deal with that stimulus. Okay, and so most of the time, reflexes are trying to protect us from something that could potentially be dangerous to us. Okay, so with a spinal reflex, and they're called spinal for a reason because these are automatic reactions to a stimulus, and these occur without conscious thought. Okay, so the brain is not involved in a reflex. That's why they're called spinal reflexes. Okay, but the caveat of that is the brain can get involved if it wants to. Right, so when your friend comes up to you and scares you, okay, there's some auditory reflex, maybe they yell, and there's some auditory reflexes involved in that that would cause you to go away from them. It's called a flexor withdrawal response. You're going to flex and move away from that sound. Okay, but the second that you realize, oh, it's just my friend, then your brain can override that reflex and stop it. Right, so the brain can get involved in reflexes, but it's not needed. Okay, the brain can override things if it needs to, but it's not necessarily needed. Okay, so it's always going to be the same thing with the reflex. We're going to have a receptor that senses some kind of stimulus. It's going to send its action potentials up a sensory neuron to the spinal cord, as we can see here. It's going to integrate in the spinal cord, and then it's going to send some kind of an effect out on a motor neuron out to skeletal muscle for the most part, as well as maybe smooth muscle and glands. Okay, so let's talk about two important ones for us today. Number one is called the stretch reflex. Number two is called the deep tendon reflex. Okay, the stretch reflex is driven by, so it says two special receptors and two reflexes. Okay, so these are the reflexes, the stretch reflex and the deep tendon reflex. The receptors here are called, this one's called the muscle spindle. This one is called the Golgi tendon organ. A lot of times we abbreviate this one as the GTO. Okay, and so the muscle spindle, we have lots of little muscle spindles inside all of our skeletal muscle. There's separate little muscle fibers that are surrounded by neurons, like you can see here. And the big thing they're detecting, is what we'll call stretch. Okay, and the key thing about stretch is it's actually telling the brain how long each muscle is, as well as the position that each muscle's in. Okay, so it's not just about pulling on both sides. It does detect that pulling, that's the stretch, but it also is constantly telling the brain, all right, how long is that muscle? How long is that muscle? How long is that muscle? Because that's how we have postural control, is by the brain knowing how long each of our muscles are. And it knows, for example, that like in a baby that's learning how to sit up, right? When they start leaning over like this, they start falling down, it's stretching all the muscles on this side of their body, right? And once that baby learns how to sit up, then the brain knows the length that all those muscles should be. And if that muscle starts getting too long, it contracts it. So the baby stays upright, right? But as they're learning that motor control, when those muscles get too long, the brain doesn't respond quick enough and they fall over, right? But that's just one example. This is happening all the time through all of our muscles in order to give us our posture and to keep us upright. Okay, so that's the muscle spindle. And then what the Golgi tendon organ detects is really what's called force, or what I prefer to call tension. Okay, and tension is really just about how hard are we pulling on both sides of that tendon. Okay, it's just how much force are we putting, how much tension are we putting on both sides of that tendon. And those are little receptors embedded inside of all of our tendons connected to our skeletal muscle. Okay, so let's talk about the first reflex here then. Okay, so the stretch reflex, this is something that you'll definitely need to know the steps of Okay, and so with any reflex, we have these exact things. We have a purpose of the reflex, what kind of receptor it is, what kind of stimulus does that receptor detect, and then all of the actions here in the pathway. Okay, so the purpose of the stretch reflex is to prevent overstretching of a muscle. Okay, the muscle spindle is the receptor used, and the stimulus detected is stretch. Okay, and so I'm going to use these the steps right here to kind of fill in the next slide to show you what's going on. Okay, so these are the steps that you need to know, and we'll go over to this slide and kind of walk through it, right? So this is that reflex that happens when somebody takes a reflex hammer and hits you on the patellar tendon on the knee, right? What happens when you hit the patellar tendon? It pulls on the patellar tendon, which pulls on the quads a little bit, and the quads rapidly stretch. Okay, so step one here, we see the muscle spindle. So we can say stretch detected. Okay, so it's detected by the muscle spindle. That's going to lead to step two, which is an action potential on an afferent neuron. All right, so that afferent neuron is going to send an action potential back to the spinal cord. Okay, and then what you see there is that neuron branches into two different branches, right? So we have 3A, which is this one right here. It has a little plus sign that shows the blue going to kind of the pink one. And so that's going to lead to an action potential on the motor neuron that's going to go to the quadriceps, right? So that's going to lead to activation of quad motor neuron, okay? And then that's going to lead to contraction of the quads. And what does our knee do when we contract our quads? Is it straight? Does it extend or flex? Put an E in if you think extend on the chat. If you put an F in if you think flex. Go ahead. E or F, extend or flex. And when our quads contract, what does our knee do? Good. Lots of E's. I like it. You guys did remember something from last semester. Good. No, that's awesome. Fantastic. Absolutely. So that's what happens, right? We stretch those quads. And then the opposite of stretching the quads is shortening them, right? And that's what we do when we contract it. So this reflex makes perfect sense. If we're worried about overstretching it, it would make sense to shorten them. And that's what contracting does. But then the other thing that makes sense is what happens in 3B right here, which is we activate an interneuron. Okay, and that's that little green one up there. The little green one is the interneuron. And what that does is that inhibits the antagonist of the stretch muscle. Okay, first one to put the antagonist of the stretch muscle in the chat wins today. I'm not sure what that means, but my kids always do. They say first one there wins, but they never really know what wins mean. But which one? What's the antagonist of the quads? There we go. Zoe got it. Oh no, Mallory got it. Yeah. No, Olivia Shaw got it. Good job, Olivia. Okay, you guys are all really close. But yeah, so the hamstrings, right? So we actually inhibit the hamstrings because if we contracted the hamstrings when we were worried about stretching the quads too much, what would happen? We would stretch the quads more, right? So it's exactly the opposite of what we want to happen if we contract the quads. So it makes perfect sense if we're worried about stretching the quads too much to contract the quads and inhibit the hamstrings so they can't contract, right? And those are the two actions here, and that's what we see. So inhibitory neuron inhibits activation, so relaxation of the antagonist, contraction of the agonist or the one being stretched, right? So that's always the stretch reflex. It happens all over the body, not just there in the quads. Okay, questions about the stretch reflex at all? Okay, good. So, and then we have these other couple reflexes that are important for you to know as well. So again, the deep tendon reflex is driven by that little sensory organ called the Golgi tendon organ, the GTO. Okay, this one doesn't protect against overstretch, it protects against overcontraction. Okay, and that's sensed by when there's too much tension on a tendon. Okay, and so what this does is the purpose is protecting against overcontraction. The stimulus is tension in the tendon, and the response that we get is that same muscle that was contracting, it gets inhibited. It it's caused to relax. Okay, example of this, remember that disease or the bacterial infection we talked about last semester, tetanus, right? That leads to super hard contraction of muscle. That completely overrides this reflex, and you can actually rip your muscles right off of your bone when you have tetanus. Okay, because that just shows you how strong your skeletal muscles are. They're always stronger than we can actually use, right? And so if we can fully activate our skeletal muscle, we can actually break our bones and rip our muscles right off of our bones. Okay, and so this Golgi tendon organ is helping us to not do that, right? It's helping us to know how much tension we can handle before it stops us from doing that. But it also gives us a reserve if we're in an emergency situation, like we need to lift something really heavy off of our child, right? Then we can use extra muscle power to do that. And there's well-documented evidence of people doing crazy things like lifting cars off of people and stuff. And a lot of that comes by completely overriding this deep tendon reflex. Okay, and then lastly, we have the flexor withdrawal reflex, which is similar to what I just talked about with the being scared, but it also happens with pain. Okay, and you guys will do something like this in the lab. We actually poke each other on the neck. Part of that reflex is an eyeball reflex, but the other part of it is you pull away from it, right? When you touch something hot, what happens is you flex all of your muscles. You flex your fingers, you flex your wrist, you flex your elbow, you flex your shoulder. All of the flexor muscles activate because you're pulling away from that pain sensation. Okay, so let's go ahead and do this last question here on the poll. And yeah, I should be able to do that. One sec. I'll go to my next poll. Okay, maybe I don't know how to go to the second poll. Share it and stop sharing. Oh, here we go. I know how to do it. Okay, here we go. Go ahead. So you're going to choose all of the answers that you think are true here. It could be more than one. Okay, so which of the following is or are true? All right, got about 30 of you. Let's give you about 15 more seconds. All right, go ahead and submit your answers there. Almost got everybody. Anybody else? Okay, there we go. I think that's everybody. Okay, so let's see what you guys said. Good, so most of you said B and C, and that's correct. Muscle spindle sense stretch, Golgi tendon organs tension. Stimulation of muscle spindle causes contraction of the stretched muscle and inhibition of the antagonist of that muscle. Stimulation of the Golgi tendon organ causes contraction. No, it causes inhibition of the contracting muscle. Okay. All right. And then, yeah, lastly, we'll just finish up with this slide. So we can have some issues with reflexes, some problems. Hyperreflexivity just means too much of a reflex. This happens when the brain, for some reason, can't control our reflex, can't stop our reflexes. So anything that slows the brain down, right? Alcohol, drugs, being distracted, or actual spinal cord damage, that connection between the brain and the spinal cord is messed up. That can lead to hyperreflexivity. Hyporeflexivity is the opposite, is when we don't have strong enough reflexes. This can happen when there's issues in the peripheral nerves, right? The nerves are starting to break down for some reason. Type 2 diabetes can lead to that, or we can actually just have damaged, torn nerves, different things like that. If we damage our nerves and we can't get that signal to the spinal cord fast enough, we can't get that activation out to the muscle fast enough to react. All right, guys, great to be with you. We'll see you again tomorrow, same time, 1:30, same Zoom link. Make sure to get that Master A&P done before 1:30 tomorrow. All right, have a great rest of your day.
Thank you.
Thank you.
Thank you.
Thank you.